with some of the techniques that I just want you to know exist right now. Okay? So there are different ways in which we can classify microorganisms. The way that we are primarily going to do it in class is through metabolic testing. And that's what we start in lab tomorrow. So, for example, is an organism catalase positive or is it catalase negative? In other words, does it have the enzyme catalase? And what we're going to try to be doing in here and in lab is tying together then what does it mean for the organism to be positive and negative with the results and the interpretation of that. We're going to go through a couple of images here of different types of techniques that we won't be able to cover in lab throughout the semester, but they are ones that I want you to know the existence of because these can commonly be used in clinical settings. And some of them are also used in research settings to identify microorganisms or determine their identity. So here is the first one. This is an image here of a test known as, well, I guess that's for you to figure out. So this is an example of a serological test, or one that includes antibodies to detect a microorganism. What is an antibody? What is it? It's what fights off infection. So we'll come back to this later on, but antibodies are specific to individual proteins, highly specific. In this case here, this is a test in which on the left you might have, or you might be testing for the ability of your antibody to detect a specific strain of bacteria. So if you wanted to, for example, distinguish O157H7 E. coli from a O26 or one of the other pathogenic forms, you might do this particular type of experiment. Excuse me. In this experiment, you take antibody, you take your bacteria, mix them together. If your microorganism is bound by the antibody, it clumps, and that's a positive result. So if, for example, let's say that these are two samples here. One has, let's say, O26 E. coli. One has O157 E. coli. Our antibody is an anti-O26 E. coli antibody. And the positive result would look like what we see on the left there. That's known as clumping, or what's the other term to describe this? And it's one of the four up there. What's that? C. Do you want to try to say it?
Agglutration?
Not too far off. Agglutination. Agglutination, or clumping. Sometimes you may hear the term hemagglutination, which is referring to red blood cell clumping. That's kind of a common sign or a common test for influenza infection. But this clumping or agglutination is one potential test that is used to identify microorganisms. Maybe that's the only. Oh, there we go. Okay. Another way that we can do this is by measuring what we call GC content. What does GC refer to? What if I included also the letters A and T? It refers to nucleotides, right? So GC, or guanine and cytosine, are two of the four nucleotides. GC content, or the percentage of guanine and cytosine in an organism's genome, is a commonly used measurement to roughly characterize. None of these techniques are perfect, but this one is kind of a very broad level way to distinguish two microorganisms from each other. Okay? We'll help to maybe separate families, but it will definitely not be able to help you distinguish one strain from another strain, or two highly related species from one another. So genome, yes, genome was the right answer. Oh, this one doesn't have the image showing up. Oh, there we go. Okay. This here is known as the 16S RNA or rDNA. What is the theme of a dichotomous key? What is the goal of any dichotomous key? Or why do we use them? Maybe is a different way to ask it. To narrow things down, right? To ask yes or no questions so that, in the case of microbiology, to narrow it down so that we can identify an unknown among a population of potential unknowns. What makes up a good question on a dichotomous key? So if you were playing Guess Who, for example, what makes up a good question?
Very specific.
Specific questions? What do you mean by that, Allison? Or give an example, if you would like.
It doesn't have many possible answers.
Good, that's great. Typically, a good question has a dichotomous key or a dichotomous question. Di means two here. It has one of two possibilities. And the best ones are ones that the answer is clearly yes or clearly no. What are weak characteristics of a question? And some of you identified these in each other's dichotomous keys that you created on VoiceThread. Opinion-based questions are definitely ones that we would consider to be weaker. What else?
If they're not necessarily like a yes or no question.
Yeah, if they're not yes or no, if it's like a yes, sometimes. Like there was, I'll give one example, and I'm not picking on this one because I think it was a particularly bad question, but because this is the one that sticks in my mind most of all. It was a question about, is the ball orange? Talking about different sports. And immediately the image that might come to your head is basketball, maybe, if you're thinking of something that's orange. But also, some of the people that were responding to it were pointing out, well, golf balls can be orange sometimes too. Or my thought was, basketballs aren't always orange. Jake's got one that's red and black. Like, that's just popular among kids now, is to have basketballs that look different than orange. So ones where the answers might be a bit more ambiguous or up in the air are ones that we would consider not to be as strong. All right? So the questions that we're trying to develop in dichotomous keys are ones that at the end of the day, it's very easy to say, yes, this is, the organism either falls under the yes or the no category. If we were to ask a question, for example, about gram staining, okay, we could ask, is the organism gram-positive? What doesn't fit nicely under the categories of gram-positive or gram-negative? Of the three types of classifications of bacteria we talked about last week on Wednesday, what doesn't fit very well under that dichotomy of gram status? Annie? The acid fast. They're considered part of gram-positives, but do they stain gram-positive? No, they don't even stain gram-negative. We have to use a different technique to detect them. So ones that have a bit of ambiguity can be a bit harder to try and work with. We're going to take the rest of today doing nothing but practicing these. Okay? Because ultimately what you and your lab partner have to do is to create one of these with the 26 unknown bacteria that are given to you. So the first worksheet that I handed out today is one that I've used in the past, and I like because it's a fairly simple one. You'll note that there's two sides to this. Okay, so there are, on this, the front page, it shows you your organisms A through K, in terms of morphology at the top. And then on the bottom half, there's a table of A through F, listing whether these organisms are positive, indicated by a plus, or negative, indicated by a minus, for various tests. and then G through K is on the back, okay?